Welcome to all of you. My today's lecture is types of RNA and ribosomes. First, I would like to introduce my topic with RNA, that is ribonucleic acid, which is one of the three major macromolecules along with DNA and proteins that are essential for all known forms of life. Like DNA, RNA is made up of a long chain of components called nucleotides. Each nucleotide consists of a nucleobase, sometimes called as nitrogenous base, a ribose sugar and a phosphate group. The sequence of nucleotides allows RNA to encode genetic information. For example, some viruses use RNA instead of DNA as their genetic material. And all organisms use messenger RNA, mRNA, to carry the genetic information that directs the synthesis of proteins. Like proteins, some RNA molecules play an active role in cells by catalyzing biological reactions, controlling gene expression, or sensing and communicating responses to cellular signals. One of these active processes is protein synthesis, a universal function whereby mRNA molecules direct the assembly of proteins into ribosomes. The process uses transfer RNA molecules to deliver amino acids to ribosomes, where ribosomal RNA links amino acids together to form proteins. The chemical structure of RNA is very similar to that of DNA. The two differences: RNA contains the sugar ribose, while DNA contains slightly different sugar deoxyribose, a type of ribose that lacks one oxygen atom. And RNA has the nucleobase uracil, while DNA contains thymine. Uracil and thymine have similar base pairing properties. Like DNA, most RNA molecules are single-stranded. Single-stranded RNA molecules adopt very complex three-dimensional structure, since they are not restricted to repetitive double helical form of double-stranded DNA. RNA is made within the living cells by RNA polymerases, enzymes which act to copy a DNA or RNA into a new RNA strand through processes known as transcription or RNA replication respectively. Now ribosomes. Ribosomes are cytoplasmic granules composed of RNA and proteins, at which protein synthesis takes place. They were first observed by Palade in the electron microscope as dense particles or granules. Upon isolation, they were shown to contain approximately equal amounts of RNA and protein. There are two types of ribosomes in the living organism: the eukaryotic 80S and prokaryotic 70S ribosomes. The subunits of 80S ribosomes are 60S and 40S units, whereas 70S have 50S and 30S subunits. Prokaryotic and eukaryotic subunits do not differ in any fundamental way. Both perform the same functions by the same set of chemical reactions. The genetic code is same in all the living organisms, and it has been demonstrated that eukaryotic ribosomes are able to translate bacterial mRNAs correctly. Eukaryotic ribosomes are much larger than that of prokaryotic ones, and most of these proteins are different. Antibiotics such as chloramphenicol inhibit bacterial but not eukaryotic ribosomal activities. Protein synthesis by eukaryotic ribosomes is inhibited by cyclohexamide. Mitochondrial and chloroplast ribosomes resemble those in bacteria. Now, the types of RNA and their description. There are three main types of RNA: mRNA, tRNA, and RNA. These are discussed as under. Messenger RNA, that is also symbolized as mRNA, it is a molecule of RNA encoding a chemical blueprint for a protein product. mRNA is transcribed from a DNA template and carries coded information to the sites of protein synthesis. Here, nucleic acid polymer is translated into a polymer of amino acids, a protein. In mRNA, as in DNA, genetic information is encoded in the sequence of nucleotides arranged into codons. Each codon encodes for a specific amino acid except the stop codon that terminates protein synthesis. This process requires two other types of RNA: transfer RNA that mediates recognition of codon and provides the corresponding amino acid, while ribosomal RNA, that is also called rRNA, is the central component of the ribosome's protein manufacturing machinery. The brief existence of an mRNA molecule begins with a transcription and ultimately ends in degradation. During its life, the mRNA molecule may also be processed, edited and transported prior to translation. Eukaryotic mRNA molecule often requires extensive processing and transport, while prokaryotic molecules do not. The short-lived, unprocessed or partially processed product is termed as premature mRNA. Once completely processed, it is termed as mature mRNA. A fully processed mRNA includes 5' cap, 2 UTR regions, coding regions, and a poly-A tail. Poly-A tail means polyadenine tail. Now we will describe these components of mRNA in detail. 5' cap. The 5' cap is a modified guanine nucleotide added to the front 5' end of the premature mRNA. This cap helps in the recognition and proper attachment of ribosomes, protection from 5' exonucleases. It is also very important for other essential processes such as splicing and transport. Coding regions. Coding regions are composed of codons which are coded and translated into proteins by the ribosomes. Coding regions begin with the start codon and ends with the stop codon. Generally, the start codon is AUG triplet and the stop codons are UAA, UAG and UGA triplets. Untranslated regions or UTRs are sections of the mRNA before the start codon and after the stop codon that are not translated, termed as 5' untranslated regions and 3' untranslated regions respectively. These regions are transcribed with the coding region and thus are exonic as they are present in the mature mRNA. Several roles in gene expression have been attributed to the untranslated regions, including mRNA stability, mRNA localization, and translational efficiency. The ability of a UTR to perform these functions depend on the sequence of UTR in different mRNAs. The stability of mRNAs may be controlled by these UTRs due to their varying affinity for RNA degrading enzymes called ribonucleases, and for the proteins that can promote or inhibit RNA degradation. Proteins that bind either 5' or 3' UTRs may affect translation by influencing the ribosomal ability to bind to the mRNA. Cytoplasmic location of mRNA is thought to be the function of 3' UTR. Proteins that are needed in a particular region of the cell can actually be translated there. In such a case, the 3' UTR may contain sequences that allow the transcript to be localized to this region for translation. Polyadenine tail. The 3' polyadenine tail is a long sequence of adenine nucleotides, often several hundred, added to the 3' end of the premature mRNA. The tail promotes export from the nucleus and translation and protects the mRNA from degradation. Transfer RNA. Transfer RNA is a small RNA molecule, usually about 73 to 95 nucleotides, that transfers a specific active amino acid to a growing polypeptide chain at the ribosomal site of the protein synthesis. It has the 3 terminal sites for amino acid attachment. This attachment is a covalent linkage which is catalyzed by an amino acyl tRNA synthetase. It also contains a 3-base region called the anticodon that can base pair to the corresponding 3-base codon region on mRNA. Each type of tRNA molecule can be attached to only one type of amino acid because the genetic code contains multiple codons that specify the same amino acid. tRNA molecules bearing different anticodons may also carry the same amino acid. The structure of tRNA can be described into its primary structure, its secondary structure, usually visualized as clover leaf structure, and its tertiary structure. All tRNAs have similar L-shaped 3D structure that allows them to fit into P and A sets of the ribosomes. The clover leaf structure becomes the 3D L-shaped structure, which is a common RNA tertiary structure. Various components of tRNA are as under. The 5' terminal phosphate group. Next is the acceptor system is a 5-7 base pair system made by the base pairing of the 5 terminal nucleotide with 3' nucleotide terminal, which contains the CCA 3' terminal group used to attach the amino acid. The acceptor system may contain non-Watson-Crick base pairing. Next is CCA tail. Is a cytosine adenine sequence of the 3' end of the tRNA molecule. This sequence is important for the recognition of tRNA by enzymes critical in translation. In most prokaryotic tRNAs and eukaryotic tRNAs, the CCA sequence is added during processing and therefore does not appear in the tRNA gene. It is involved in the process of amino acylation. That is, a specific amino acid is added to the CCA 3' end of the tRNA by the formation of covalent bond catalyzed by the enzymes amino acyl tRNA synthetases. There is normally a single amino acyl tRNA synthetase for each amino acid, despite the fact that there can be more than one tRNA and more than one anticodon for an amino acid. Recognition of the appropriate tRNA by the synthetases is mediated solely by the anticodon, but the acceptor system often plays a prominent role. The DRM is a 4 base pair system ending in a loop that often contains dihydrouridine. The anticodon RM is a 5 base pair system whose loop contains the anticodon. The anticodon is a unit made up of 3 nucleotides that corresponds to the 3 bases of the codon on the mRNA. Each tRNA contains a specific anticodon triplet sequence that can base pair to one or more codons for an amino acid. For example, the codon for lysine is AAA. The anticodon of that lysine tRNA might be UUU. Some anticodons can pair with more than one codon due to the phenomenon known as Weibull base pairing. In this type of pairing, only 2 bases of the triplet determines the specificity for the amino acid to be decoded, whereas the 3rd base of the triplet can be any of the 4 bases. For example, the amino acid glycine is coded by any of these codon sequences like GGU, GGC, GGA and GGG. The tRM is a 5 base pair system containing the sequence T-Psi RM, where Psi is a pseudouridine. Bases that have been modified, especially by methylation, occurs at several positions throughout the tRNA. Now, ribosomal RNA. Ribosomal RNA or rRNA is the RNA component of the ribosome, the protein manufacturing organelle of all the living cells. Ribosomal RNA provides a mechanism for decoding mRNA into amino acids and interacts with tRNAs during translation by providing peptidal transferase activity. The tRNAs bring the necessary amino acids corresponding to the appropriate mRNA codon. Now, non-coding RNA. There are certain other RNAs in addition to main RNAs. These are known as non-coding RNAs. These are not translated into proteins. Less frequently used synonyms for non-coding RNAs are non-protein coding mRNAs, non-messenger RNAs, small non-messenger RNA and functional RNA. The term small RNA or sRNA is often used for small bacterial ncRNAs. The DNA sequences from which a non-coding RNA is transcribed, often called as RNA gene or non-coding gene. The structure and function of ribosomes. Ribosomes are tiny particles about 200 angstrom. Each ribosome is composed of both proteins and RNA. In fact, it has approximately 37 to 62% RNA and rest are made up of proteins. The RNA present in the ribosomes are obviously called ribosomal RNA. and they are produced in the nucleolus, which is a prominent globular structure in the nucleus. The ribosomes fall into two categories: those that are free to roam in the cytoplasm and those that are attached with the membranes of endoplasmic reticulum, thus causing a rough surface. Although the two kinds of ribosomes play similar roles in translating mRNA to produce proteins, the ribosomes in the cytoplasm allows its proteins to roam about freely, while the bound ribosomes transfer their functional proteins into the endoplasmic reticulum. In addition, ribosomes are located within the mitochondria and in the chloroplast. These are spherical particles composed of two subunits, a large and a small. In eukaryotes, the coefficient of ribosomes are 80s, which are divided into 60s for the large and 40s for the smaller subunits. The 60s contains 28s RNA with a small fragment 5.8s that is attached non-covalently and can be released upon heating, and very small 120 nucleated 5s RNA. Whereas the 40s subunit, it has only a single 18s RNA. In prokaryotes, however, the large and the smaller subunits are 50s and 30s, making a total of 70s respectively. The 50s has two types of RNAs, a 23s and a 5s. It also has 32 different proteins. On the other hand, the 30s contains a single 16s RNA and 21 different types of proteins. The 50s and 30s refers to the sedimentation coefficient of the two subunits. This coefficient is a measure of the speed with which the particles sediment through a solution which spun in an ultracentrifuge. Thus, the particles with larger coefficient would centrifuge and settle much faster since it has more mass than the particles with a smaller coefficient. During protein synthesis, ribosomes line up along the mRNA and form a polysome, also called a polyribosome. The mRNA is aligned in the gap between the two ribosomal subunits. It is possible that the nascent peptide chain grows through a channel or groove in the large ribosomal subunit. Up to 30 ribosomes can attach to one strand of mRNA to form amino acid chains, thus leading to protein formation. Ribosomes act as a backbone during translation in the protein synthesis. Each ribosome contains the P site and A site for tRNA binding. The ribosome molecules allow growing polypeptide chain to emerge from the back of the surface. Thus, it is situated perpendicular to mRNA chain. Ribosomes have a tertiary structure and make up a large part of the cells in many species, which leads to protein manufacturing. For example, in E. coli bacteria, they make up about 1/4 of the total cell mass. Now, the students will describe the function of these ribosomes. The ribosomes play a very important role in protein synthesis, which is the process by which the proteins are made from the amino acids. Without the ribosomes, the message would not be read. Thus, proteins could not be produced. Therefore, ribosomes play a very important role in protein synthesis. The ribosomes are the primary agents in the process of translating the mRNA into the specific amino acid chain, which consists of two subunits. These subunits are made up of ribosomal RNA, together contains up to 82 specific proteins assembled in a precise sequence. The ribosomal subunits must be put together in an extremely precise position and sequence. This assembled ribosome displays a series of small grooves, tunnels, and platforms where the function of protein synthesis occurs. There are the active sites, each dedicated to one of the tasks required for translation of mRNA into proteins. Proteins being synthesized for export out of the cell are made up of ribosomes attached to rough endoplasmic reticulum. In contrast, proteins for use by the cell are generally made in the cytoplasm by free ribosomes. Several of these free ribosomes may attach to a single mRNA molecule. giving rise to the polyribosome or polysome. Protein synthesis takes place on the polyribosomes, where ATS ribosome associate with an mRNA coding for a given protein. The number of ribosomes associated in the polysomal chain depends upon the size of mRNA. This is also associated with the size of proteins that is being synthesized. Transfer RNAs are also bound to the ribosomes. There are quite a few factors involved in the formation of initiation complex. These include GTP, methionine tRNA, and initiation codon in mRNA, ATS ribosomes, and 3 protein factors. The process of protein synthesis begins with the capture of the tRNA, which is carrying an amino acid, by an initiation factor. This binds to the small ribosomal subunits, which occupies one of the active sites in the ribosomes, the P protein site. This initiation complex recognized and binds to the 5' end of the mRNA molecule and slides down to the initiation codon, which is always an AUG sequence of an amino acid. The large subunits of ribosomes now join the complex. tRNA is now brought into the ribosome by the elongation factor. If the anticodon of the tRNA pairs with the next codon of the message, the tRNA occupies a site, acceptor site of the ribosome. This positions the second amino acid adjacent to the initiation methionine. Then an enzyme, peptidyl-transferase, which is the part of large ribosomal subunits, mediate the separation of first amino acid and its tRNA, and the formation of a peptide bond between the initial methionine and the amino acid is formed. The P site is now occupied by an uncharged tRNA molecule. The ribosome will now move down the mRNA by one codon, a process known as translocation. This movement shifts the growing polypeptide chain to the P position and results in an empty A site, where a new charged tRNA can enter and pair by forming a hydrogen bond between the codons and the anticodon. Now, the uncharged tRNA that previously occupies the P position is booted out of the ribosome and will be recharged and recycled by the cell. The energy needed for this process is supplied by the hydrolysis of guanosine triphosphate (GTP). This process then continues along the length of mRNA until the first codon is encountered. At that point, the action of termination factor releases the completed protein from the last tRNA and the ribosome dissociates into its component parts. Dear students, this way we have completed our lecture. I hope all of you have liked it. Thank you.